Our fourth SoftChalk lesson in Unit 1, Lecture 4 deals with bacterial ribosomes. So let's look at our fundamental statements and detailed learning objectives for this SoftChalk lesson. Bullet points: ribosomes are composed of ribosomal RNA or rRNA and protein. Bacterial ribosomes are composed of two subunits with sedimentation rates of 50S and 30S, as opposed to the 60S and 40S ribosomal subunits found in eukaryotic cells. Ribosomes function as a workbench for protein synthesis, where they receive and translate genetic instructions for the formation of specific proteins. During translation, specific transfer RNA molecules pick up specific amino acids, transfer those amino acids to the ribosome, and insert them in the proper place according to the messenger RNA message. In bacteria, transcription and translation are coupled. Many antibiotics bind to either a 30S or 50S ribosomal subunit of bacterial ribosomes and thus interfere with translation, causing faulty protein synthesis in the bacteria. And looking at our detailed learning objectives, you should be able to describe the structure and chemical composition of bacterial ribosomes and state the function of ribosomes. In terms of protein synthesis, briefly describe the processes of transcription and translation, and state in a general sense how antibiotics like neomycin, tetracycline, doxycycline, erythromycin, and azithromycin affect bacterial growth. So as we learned when we compared pro and eukaryotic cells in an earlier SoftChalk lesson, and as you know from your biology course, ribosomes are composed of ribosomal RNA or rRNA and protein. Now, prokaryotic cells or bacteria have three types of ribosomal RNA: a 16S ribosomal RNA, a 23S ribosomal RNA, and a 5S ribosomal RNA. And like transfer RNAs, ribosomal RNAs have a lot of intrahydrogen bond between complementary nucleotide bases in the same strand to form a complex three-dimensional folded structure. So bacterial ribosomes, like all ribosomes, are composed of two subunits, and the two subunits have sedimentation rates of 50S and 30S. We described the Sedberg unit, or unit of sedimentation, back under our prokaryotic and eukaryotic SoftChalk lesson. Now, the 30S ribosomal subunit contains a 16S ribosomal RNA and 21 proteins, while the 50S subunit contains a 5S and a 23S ribosomal RNA and 31 proteins. And the two subunits come together during protein synthesis to form a 70S ribosome that's about 25 nanometers in diameter. Remember, a nanometer is a billionth of a meter. And a typical bacterium can have as many as 15,000 ribosomes. And in Figure 1, we see an electron micrograph of Streptococcus pneumoniae, where we can see the chromosome in the nucleoid area, and notice how that is surrounded by ribosomes. All the black dots you see in this electron micrograph are ribosomes. And as the DNA is being transcribed into messenger RNA, it's being fed directly into ribosomes for translation. Now, of course, in function, as you've always learned, ribosomes function as a workbench for protein synthesis. And what that really means is that they receive and translate genetic instructions for the formation of specific polypeptides or proteins. So during protein synthesis, messenger RNA is going to be attaching to the 30S subunit, being fed into it, where then amino acid carrying transfer RNAs read the message as they attach to the 50S subunit. So as we learned under the chromosome is the genetic material of bacteria. And of course, our dogma here is that genes located along the DNA are transcribed into RNA molecules, messenger RNA, transfer RNA, and ribosomal RNA molecules. And most of the genes are transferred or transcribed into messenger RNA or mRNA. And then messenger RNA is translated into proteins at the ribosomes. And in bacteria, transcription and translation are coupled together. Unlike eukaryotic cells where transcription occurs in the nucleus and then translation occurs outside the nucleus. So in bacteria, RNA polymerase binds to a 30S ribosomal subunit of a prokaryotic ribosome to form a transcription and translation machine that's called an expressome. And as the DNA is being unwound and being transcribed into complementary messenger RNA by RNA polymerase, the messenger RNA is being fed into the translational center of the ribosome, where it's being translated into a polypeptide or protein. So a brief review of our terms here, transcription and translation. We covered these in quite a bit on your take-home exam that covered the molecular genetics portion of your biology prerequisite course, but let's review the definitions. So during transcription, transcribe means to copy, where RNA is making a complementary copy of a gene. So ribonucleic acid, or RNA, is synthesized by complementary base pairing of ribonucleotides with deoxyribonucleotides on the gene so that it makes a complementary copy of that gene. Now, although there's genes present on both strands of DNA, only one strand is transcribed for any given gene. Following transcription of genes into messenger RNA, 30S and 50S ribosomal subunits attached to the messenger RNA, and then transfer RNA inserts the correct amino acid to form the right polypeptide or protein through the process called translation. So during transcription, the gene is copied into complementary messenger RNA in terms of protein synthesis. Then during translation, which means to translate the messenger RNA message, specific transfer RNAs pick up specific amino acids. They transfer those amino acids to the ribosome, and they insert them in the proper place according to the messenger RNA message. And this is done by the anticodon portion of the transfer RNA complementary base pairing with the codon along the messenger RNA. So remember, an anticodon is a series of 3 ribonucleotide bases at the bottom of a molecule transfer RNA that is complementary to a messenger RNA codon. And the anticodon of the transfer RNA forms hydrogen bonds with the codons along the messenger RNA, and that's how the right amino acid is inserted in the right place. And of course, complementary base pairing again refers to certain nucleotide bases forming hydrogen bonds with others. In RNA, uracil pairs with adenine and guanine pairs with cytosine. There is no thymine in RNA. And this animation here kind of summarizes the translation process. So let's review that a little bit. So we see here that a messenger RNA is being transcribed off the DNA. So these are nucleotide bases complementary to those of a gene in the DNA. At the beginning of the messenger RNA is a ribosomal binding site indicated by X's, where a 30S ribosomal subunit can first attach. So to form what's called the initiation complex, the messenger RNA is fed into the 30S subunit of the ribosome and binds by the ribosome binding portion. And then the first transfer RNA, which is always complementary to the start codon, AUG, on the messenger RNA, lines up. And it has a complementary anticodon, UAC, because uracil pairs with adenine, cytosine pairs with guanine. At that point, a 50S subunit attaches to the initiation complex, and we're ready for translation to occur. So we have 3 sites on the bacterial ribosome. The A site on the right-hand side here accepts new transfer RNA. So that's where new transfer RNAs are going to come in and line up opposite the codon under the A site. The P site is where the previous transfer RNA and the growing amino acid chain is being held. P stands for peptide, so the growing peptide chain is being held there. And the E site is where the transfer RNA exits the ribosome after the amino acid on the transfer RNA in the P site attaches to the new one on the A site, as we'll see happening here. So the next codon to be read is CGC, shown here under the A site on the 50S subunit. And if you look that up in your codon sheet, that codes for the amino acid arginine. And the reason for that is that the only transfer RNA that can complementary base pair with that is one that has a GCG anticodon. And a transfer RNA with a GCG anticodon can only carry the amino acid arginine. So as we see here, a transfer RNA with an anticodon GCG will hydrogen bond with a CGC codon, and that inserts arginine in the proper place. At this point, as the messenger RNA moves along, prior to that, the amino acid comes off of the transfer RNA at the P site and forms a peptide bond with the newest amino acid, in this case arginine, located on the transfer RNA at the A site. And there's an enzyme called peptidyltransferase that's part of the ribosome that allows this to happen. Now at that point, the messenger RNA moves down so the next codon can be read, and the old transfer RNA that was on the P site exits to the E site. And that opens up the A site, so a new transfer RNA can read the next codon, which is GGA. And that is the codon for the amino acid glycine. So that codes for glycine because the only transfer RNA that can line up there is one that has a complementary anticodon, CCU. And that transfer RNA can only carry the amino acid glycine. So the anticodon of that transfer RNA with CCU hydrogen bonds with GGA, inserting glycine in the proper place. At this point, the amino acids sitting on the transfer RNA in the P site move and form a peptide bond to the newest amino acid, in this case glycine, found at the A site. At that point, as the messenger RNA moves down the ribosome, the old transfer RNA at the P site exits through the E site, and that opens up the A site to read the next codon, which in this case is UCC. And if you look that up on your codon sheet, that codes for serine. And it codes for serine because the only transfer RNA that can line up there is one that has a complementary anticodon, AGG. And a transfer RNA with an AGG anticodon can only carry the amino acid serine. So now the transfer RNA with the AGG anticodon hydrogen bonds with its complementary UCC on the codon, inserting serine. Now the growing peptide chain on the transfer RNA to P site will form a peptide bond with the newest amino acid, in this case serine, on the transfer RNA at the A site. So each time one more amino acid is being added to the growing polypeptide chain. And as the messenger RNA moves down, the old transfer RNA at the P site that's now uncharged moves to the E site where it exits the ribosome, and that opens up a new codon to be read at the A site, which is CCC. And that codes for the amino acid proline. And only a proline-carrying transfer RNA has a complementary anticodon, GGG. So the transfer RNA with a GGG anticodon hydrogen bonds with a CCC codon, inserting proline in its proper place. Now the growing peptide chain forms a peptide bond with the newest amino acid on the transfer RNA at the A site as it leaves the transfer RNA at the P site. And then as the messenger RNA moves down, The old transfer RNA from the P site exits at the exit site, opening up the A site to read the next codon, ACC, which codes for threonine. And only a threonine containing amino acid will have a complementary anticodon, in this case UGG, that can hydrogen bond with the ACC codon, so that's why ACC codes for codon. Only a transfer RNA carrying threonine can attach to that codon. Now again, the growing polypeptide chain on the P site forms a peptide bond with the newest amino acid, in this case threonine, at the A site, leaving the old transfer RNA at the P site. Then as the messenger RNA slides down, the old transfer RNA at the P site moves to the E site, where it exits and can be recycled, opening up the reading of the last codon. And the last codon in this short message is UGA. And if you look at your codon sheet, that's one of our 3 stop codons. So there is no corresponding transfer RNA that has an anticodon complementary to UGA. So no amino acids can be inserted here. And the ribosomal subunits come off and the protein has been released. And during that process, the secondary and tertiary structure of the protein are formed and it is released in its 3-dimensional functional form. And there's a lot of good YouTube movies on transcription and translation. I've included several of those here if you want to watch them. And there's a little self-check you can do on that section. Now, one of the ways we can use antibiotics to inhibit or kill bacteria is we take chemicals that will bind to something found in a prokaryotic cell that a eukaryotic cell doesn't have, or at least has a different form of that structure. And remember, prokaryotic ribosomes are different from eukaryotic ribosomes. They carry out the same function, but they're composed of different ribosomal RNAs, different proteins, they have different sedimentation rates, and so they have different shapes even though they carry out similar functions. So there are drugs that will bind to a prokaryotic ribosome, but they wouldn't bind to a eukaryotic ribosome because they are structurally different due to the different ribosomal RNAs and the different proteins found in the ribosomes. So many of our antibiotics alter bacterial ribosomes, and they therefore interfere with translation of the message. Remember, ribosomes are involved in translation, not transcription. And of course, that causes faulty protein synthesis in the bacterium. And where the drug binds to the ribosome determines how translation is affected. And we're going to look at a number of examples of this later on in Unit 2 when we get into chemical control of bacteria. But let's look at a couple of examples here, starting with the tetracyclines. Antibiotics like tetracycline, doxycycline, minocycline, and such. Now, these drugs bind reversibly to the 30S ribosomal subunit. Binding reversibly means they can attach and they can come off, and they can attach and they can come off. And as long as they're attached, they're blocking translation. So drugs that bind reversibly to ribosomes are said to be static in action. They inhibit growth, but they don't always kill the organism. Now, when the drug binds to the ribosomal subunit, that distorts the subunit in a way that the anticodons of the charge transfer RNAs can't align properly with the codons of the messenger RNA. And we can see that in our next animation here. So here we see the messenger RNA has attached to the 30S subunit. The initiation complex is formed. The 50S subunit has attached. And here's our drug tetracycline in green. So as we said, the tetracycline binds to a 30S ribosomal subunit, and it binds in the location that's under the site for the A site of the 50S subunit. But by the drug binding to the 30S ribosomal subunit here, it prevents the transfer RNAs that are coming in from getting close enough to form hydrogen bonds with the codon. So as a result, the transfer RNA doesn't insert any amino acids. It can't form hydrogen. The anticodon of the transfer RNA can't hydrogen bond with the codon on the messenger RNA, as we see here. So again, the GCG can't get close enough to hydrogen bond to the CGC, so as a result, no amino acids are inserted. So transfer RNAs can keep coming in and try to hydrogen bond, but they can't. So once that drug binds, no more amino acids are inserted and the protein is not made. So in that case, it's preventing the transfer RNAs from being able to form hydrogen bonds with the messenger RNAs. That is, it prevents the anticodons from pairing with the codons. Now, the macrolides are another common group of antibiotics, things like erythromycin, azithromycin, clarithromycin, and such. And these bind reversibly to the 50S subunit. And they can inhibit protein synthesis a couple of ways, we think. Some can inhibit the elongation of the protein by preventing the enzyme peptidyltransferase from forming peptide bonds between the amino acids. Now, the peptidyltransferase that forms the peptide bonds between the amino acids is actually a part of the ribosome located where the peptide bonds are going to form, and the drug binds to that area, thus blocking the enzyme peptidyltransferase, so no peptide bonds are formed. The amino acids are never joined together, and that prevents protein synthesis. Others may prevent the peptidyltransferase from moving from the A site to the P site, as we'll see in the next animation coming up. But let's look at the first one here, where they are binding to peptidyltransferase, preventing peptide bond formation. So again, there's our messenger RNA, a 30S subunit attaches, a transfer RNA complementary to the start codon forms an initiation complex, the 50 subunit attaches, and we're ready to go. So here's a macrolide, and as we see, this binds to the 50S subunit, and it binds right where the enzyme peptidyltransferase is located. So now, the next codon to be read is CGC, and so a transfer RNA with a complementary anticodon, GCG, forms hydrogen bonds, but no peptide bonds form between the amino acids because the peptidyltransferase is blocked. So in this case, the transfer RNAs can line up carrying amino acids, but no peptide bond forms and no proteins are formed. And then in some cases, certain macrolides we think might bind to the 50S subunit and block translation by preventing the peptidyl transfer RNA, the transfer RNA in the P site from moving. And so it prevents the transfer RNA in the A site from moving to the P site. And let's take a look at that happening. So it's binding to a 50S subunit. Now remember, once the amino acid on the P site has attached to the newest amino acid on the transfer RNA to the A site, then as the messenger RNA moves, that moves the ribosome so that the next codon can be read. But what happens here is the drug interferes with that process so that the transfer RNA on the A site doesn't move to the P site once it's empty. And because that can't move, it basically gets knocked off the ribosome and again prevents any further elongation of the protein. So you see there the transfer RNA didn't move from the A site to the P site. So again, the transfer RNAs can line up, anticodons pairing with codons, but the transfer RNA at the A site never moves to the P site, and so no amino acids are inserted. So there's a couple ways that show how antibiotics can bind to a 30S or 50S ribosomal subunit and interfere with translation. And we'll look at some additional ways later on in Unit 2. But this all depends on where the antibiotic binds to the ribosome. It interferes with whatever is supposed to happen at that particular site or location. And there's a self-quiz you can do on bacterial ribosomes.